Hey guys, so this week, changing things up a little bit, we're going to watch two videos. We're going to watch a Kirgistat video, which means in a nutshell. They're a YouTube channel based in Germany that makes really, really well done animated videos that go into awesome detail about biological subjects, usually biology. But in this case, they're going to be talking about ethics and genetics and CRISPR and specialized babies and gene editing and things like that. And so we're going to watch their video first to give us sort of a synopsis of what the big questions are. And then we're going to switch gears and switch over to George Church. So George Church is a Harvard professor at Harvard Medical School, and he's one of the world experts on gene editing. And which has sort of made him the de facto leader of gene editing ethics. And he's going to sort of talk about, his talk is like the perils and promises of gene editing. And so it's a talk that he gave that was simulcast. And so you're going to see what a typical lecture looks like at Harvard, typical seminar. But it should be really, really good. I like it a lot. He makes one reference. I have a little text to it. It's a 2018 video. So he makes one reference to the Hawaiian guy who pushed the wrong button. That is a reference. In case you don't remember, there was a guy who worked at the Hawaii National warning system, and they accidentally pushed the button that said there was a missile incoming, from Russia or something. That was an accident, so he references that. But anyway, George Church. And so pay close attention because this is definitely one of the leading experts. And what I want you to pay attention to as well is how much he thanks the other people who have worked either underneath him or for him or with him in collaboration. So let's get to it.
Imagine you were alive back in the 1980s and were told that computers would soon take over everything from shopping to dating and the stock market. That billions of people would be connected via a kind of web. That you would own a handheld device orders of magnitude more powerful than supercomputers. It would seem absurd, but then all of it happened. Science fiction became our reality and we don't even think about it. We're at a similar point today with genetic engineering. So let's talk about it. Where it came from, what we're doing right now, and about a recent breakthrough that will change how we live and what we perceive as normal forever. Humans have been engineering life for thousands of years. Through selective breeding, we strengthened useful traits in plants and animals. We became very good at this, but never truly understood how it works, until we discovered the code of life, deoxyribonucleic acid, DNA, a complex molecule that guides the growth, development, function, and reproduction of everything alive. Information is encoded in the structure of the molecule. 4 nucleotides are paired and make up a code that carries instructions. Change the instructions, and you change the being carrying it. As soon as DNA was discovered, people tried to tinker with it. In the 1960s, scientists bombarded plants with radiation to cause random mutations in the genetic code. The idea was to get a useful plant variation by pure chance. Sometimes it actually worked too. In the 70s, scientists inserted DNA snippets into bacteria, plants, and animals to study and modify them for research, medicine, agriculture, and for fun. The earliest genetically modified animal was born in 1974, making mice a standard tool for research, saving millions of lives. In the 80s, we got commercial. The first patent was given for a microbe engineered to absorb oil. Today, we produce many chemicals by means of engineered life, like life-saving clotting factors, growth hormones, and insulin, all things we had to harvest from the organs of animals before that. The first food modified in the lab went on sale in 1994. The Flavor Saver tomato, a tomato given a much longer shelf life via an extra gene that suppressed the buildup of a rotting enzyme. But GM food and the controversy surrounding them deserve a video of their own. In the 1990s, there was also a brief foray into human engineering. To treat maternal infertility, babies were made that carried genetic information from 3 humans, making them the first humans ever to have 3 genetic parents. Today, there are super muscled pigs, fast-growing salmon, featherless chickens, and see-through frogs. On the fun side, we made things glow in the dark. Fluorescent zebrafish are available for as little as $10. All of this is already very impressive, but until recently, gene editing was extremely expensive, complicated, and took a long time to do. This has now changed with a revolutionary new technology now entering the stage: CRISPR. Overnight, the costs of engineering have shrunk by 99%. Instead of a year, it takes a few weeks to conduct experiments, and basically everybody with a lab can do it. It's hard to get across how big a technical revolution CRISPR is. It literally has the potential to change humanity forever. Why did this sudden revolution happen, and how does it work? Bacteria and viruses have been fighting since the dawn of life. So-called bacteriophages or phages hunt bacteria. In the ocean, phages kill 40% of them every single day. Phages do this by inserting their own genetic code into the bacteria and taking them over to use them as factories. The bacteria try to resist, but fail most of the time because their protection tools are too weak. But sometimes bacteria survive an attack. Only if they do so can they activate their most effective antivirus system. They save a part of the virus DNA in their own genetic code in a DNA archive called CRISPR. Here, it's stored safely until it's needed. When the virus attacks again, the bacterium quickly makes an RNA copy from the DNA archive and arms a secret weapon, a protein called Cas9. The protein now scans the bacterium's insides for signs of the virus invader by comparing every bit of DNA it finds to the sample from the archive. When it finds a 100% perfect match, it's activated and cuts out the virus DNA, making it useless, protecting the bacterium against the attack. What's special is that Cas9 is very precise, almost like a DNA surgeon. The revolution began when scientists figured out that the CRISPR system is programmable. You can just give it a copy of DNA you want to modify and put the system into a living cell. If the old techniques of genetic manipulation were like a map, CRISPR is like a GPS system. Aside from being precise, cheap, and easy, CRISPR offers the ability to edit live cells, to switch genes on and off, and target and study particular DNA sequences. It also works for every type of cell: microorganisms, plants, animals, or humans. But despite the revolution CRISPR is for science, it's still just a first-generation tool. More precise tools are already being created and used as we speak. In 2015, scientists used CRISPR to cut the HIV virus out of living cells from patients in the lab, proving that it was possible. Only about a year later, they carried out a larger-scale project with rats that had the HIV virus in basically all of their body cells. By simply injecting CRISPR into the rats' tails, they were able to remove more than 50% of the virus from cells all over the body. In a few decades, a CRISPR therapy might cure HIV. And other retroviruses, viruses that hide inside human DNA like herpes, could be eradicated this way. CRISPR could also defeat one of our worst enemies, cancer. Cancer occurs when cells refuse to die and keep multiplying while concealing themselves from the immune system. CRISPR gives us the means to edit your immune cells and make them better cancer hunters. Getting rid of cancer might eventually mean getting just a couple of injections of a few thousand of your own cells that have been engineered in the lab to heal you for good. The first clinical trial for a CRISPR cancer treatment on human patients was approved in early 2016 in the US. Not even a month later, Chinese scientists announced that they would treat lung cancer patients with immune cells modified with CRISPR in August 2016. Things are picking up pace quickly. And then there are genetic diseases. There are thousands of them, and they range from mildly annoying to deadly or entail decades of suffering. With a powerful tool like CRISPR, we may be able to end this. Over 3,000 genetic diseases are caused by a single incorrect letter in your DNA. We are already building a modified version of Cas9 that is made to change just a single letter, fixing the disease in the cell. In a decade or two, we could possibly cure thousands of diseases forever. But all of these medical applications have one thing in common. They are limited to the individual and die with them, except if you use them on reproductive cells or very early embryos. but CRISPR can and probably will be used for much more: the creation of modified humans, designer babies, and will mean gradual but irreversible changes to the human gene pool. The means to edit the genome of a human embryo already exist, though the technology is still in its early stages, but it has already been attempted twice. In 2015 and 2016, Chinese scientists experimented with human embryos and were partially successful on their second attempt. They showed the enormous challenges we still face in gene editing embryos, but also that scientists are working on solving them. This is like the computer in the 70s. There will be better computers. Regardless of your personal take on genetic engineering, it will affect you. Modified humans could alter the genome of our entire species because their engineered traits will be passed on to their children and could spread over generations, slowly modifying the whole gene pool of humanity. It will start slowly. The first designer babies will not be overly designed. It's most likely that they will be created to eliminate a deadly genetic disease running in a family. As the technology progresses and gets more refined, more and more people may argue that not using genetic modification is unethical because it condemns children to preventable suffering and death and denies them the cure. But as soon as the first engineered kid is born, a door is opened that can't be closed anymore. Early on, vanity traits will mostly be left alone. But as genetic modification becomes more accepted and our knowledge of our genetic code enhances, the temptation will grow. If you make your offspring immune to Alzheimer, why not also give them an enhanced metabolism? Why not throw in perfect eyesight? How about height or muscular structure? Full hair? How about giving your child the gift of extraordinary intelligence? Huge changes are made as a result of the personal decisions of millions of individuals that accumulate. This is a slippery slope. Modified humans could become the new standard. But as engineering becomes more normal and our knowledge improves, we could solve the single biggest mortality risk factor: aging. 2/3 of the 150,000 people who die today will die of age-related causes. Currently, we think aging is caused by the accumulation of damage to our cells, like DNA breaks and the systems responsible for fixing those wearing off over time. But there are also genes that directly affect aging. A combination of genetic engineering and other therapy could stop or slow down aging, maybe even reverse it. We know from nature that there are animals immune to aging. Maybe we could even borrow a few genes for ourselves. Some scientists even think biological aging could be something that eventually just stops being a thing. We would still die at some point, but instead of doing so in hospitals at age 90, we might be able to spend a few thousand years with our loved ones. Research into this is in its infancy, and many scientists are rightly skeptical about the end of aging. The challenges are enormous, and maybe it is unachievable. But it is conceivable that people alive today might be the first to profit from effective anti-aging therapy. All we might need is for someone to convince a smart billionaire to make it their next problem to solve. On a bigger scale, we certainly could solve many problems by having a modified population. Engineered humans might be better equipped to cope with high-energy food, eliminating many diseases of civilization like obesity. In possession of a modified immune system, with a library of potential threats, we might become immune to most diseases that haunt us today. Even further into the future, we could engineer humans to be equipped for extended space travel and to cope with different conditions on other planets, which would be extremely helpful in keeping us alive in our hostile universe. Still, a few major challenges await us, some technological, some ethical. Many of you watching will feel uncomfortable and fear that we will create a world in which we will reject non-perfect humans and pre-select features and qualities based on our idea of what's healthy. The thing is, we are already living in this world. Tests for dozens of genetic diseases or complications have become standard for pregnant women in much of the world. Often, the mere suspicion of a genetic defect can lead to the end of a pregnancy. Take Down syndrome, for example, one of the most common genetic defects. In Europe, about 92% of all pregnancies where it's detected are terminated. The decision to terminate a pregnancy is incredibly personal, but it's important to acknowledge the reality that we are pre-selecting humans based on medical conditions. There is also no use in pretending this will change, so we have to act carefully and respectfully as we advance the technology and can make more and more selections. But none of this will happen soon. As powerful as CRISPR is, and it is, it's not infallible yet. Wrong edits still happen, as well as unknown errors that can occur anywhere in the DNA and might go unnoticed. The gene edit might achieve the desired result, disabling a disease, but also might accidentally trigger unwanted changes. We just don't know enough yet about the complex interplay of our genes to avoid unpredictable consequences. Working on accuracy and monitoring methods is a major concern as the first human trials begin. And since we've discussed a possible positive future, there are darker visions too. Imagine what a state like North Korea could do if they embraced genetic engineering. Could a state cement its rule forever by forcing gene editing on their subjects? What would stop a totalitarian regime from engineering an army of modified super soldiers? It is doable in theory. Scenarios like this one are far, far off into the future, if they ever become possible at all. But the basic proof of concept for genetic engineering like this already exists today. The technology really is that powerful. While this might be a tempting reason to ban genetic editing and related research, that would certainly be a mistake. Banning human genetic engineering would only lead to the science wandering off to a place with jurisdiction and rules that we're uncomfortable with. Only by participating can we make sure that further research is guided by caution, reason, oversight, and transparency. Do you feel uncomfortable now? Most of us have something wrong with them. In the future that lies ahead of us, would we have been allowed to exist? The technology is certainly a bit scary, but we have a lot to gain, and genetic engineering might just be a step in the natural evolution of intelligent species in the universe. We might end disease. We could extend our life expectancy by centuries and travel to the stars. There's no need to think small when it comes to this topic. Whatever your opinion on genetic engineering, the future is approaching no matter what. What has been insane science fiction is about to become our new reality, a reality full of opportunities and challenges.
He's one of the most renowned scientists in the world, as you are well aware. He's a professor of genetics at Harvard Medical School, professor of health sciences and technology at Harvard and MIT, director of the Personal Genome Project, and internationally recognized as a pioneer in genome sequencing and synthetic biology. He has co-authored more than 450 papers and holds more than 100 patents. Among a lengthy list of honors, did you know that last year Dr. Church was named one of the 100 most influential people by Time magazine? As Stephen Colbert said recently, Dr. Church's contribution to genetic research and the imagination he brings to its application may change the entire world and our experience of life itself. We are tremendously honored to have him here today. Thank you, and please join me in welcoming him.
Thank you very much. Everybody hear me? Yeah, in the back there? Yeah. So I sort of feel like looking at all the people here, like I'm the guy in Hawaii that pressed the wrong button. And we're all hunkered down in the basement. But anyway, I do believe there has been a little bit of hype both on the promise side and on the peril side. So I'm going to try to maybe de-emphasize the precise exactly what the title is about, but you'll see, and try to hit some kind of balance. This is my thank you slide. It's also my conflict of interest slide. And I'll have a whole bunch of thank yous along the way. You'll see that it's pretty much just a thank you talk. That's it. Here's thank you to pged.org, which is, one of the few and best, I think, organizations for educating the people, not just the already well-educated people in this room, including Congress, high schools, screenwriters, and so forth. And I'm just very indebted to the work that they've done for now over a decade. And then I want to say thank you to Heidi Ledford for her article on the unsung heroes. In particular, I'd like to thank first and middle authors who often get forgotten in conversations like the one we're going to have today. And so here is this slide entirely on first and middle authors that I think made big contributions to gene editing, including CRISPR up there in the upper right-hand corner. And we, lest we forget, there was pre-CRISPR that actually helped us be ready for CRISPR and things beyond it, and those include zinc finger nucleases, homing into nucleases, and so forth. And I think the really pioneer of all this was Bernard Dujon. Unfortunately, I didn't find my slide of him when he was when I was a graduate student with him. But he not only found homing in the nucleus, which was arguably the first of the programmable nucleases in 1976, but also gene drives, which I'll mention briefly. And his and my mentor at that time, Piotr Slimsky, both in Paris, or a suburb of Paris, Chiffre-Vert, invented the term guide RNA back in '79. But all these people deserve much more time than we have here. I want to get on to the lack of promise and lack of peril topic. So these would constitute, none of these slides, by the way, are comprehensive, but these would constitute some of the promises in alphabetical order. No other particular order, that we can reverse aging, we can increase biodiversity that is imperiled, we can reverse climate change, we can bring back the dead. Why not? I mean, we're in the promises. And we can make things extinct. We can be extinct and make them extinct, hopefully malaria, Lyme disease, HIV, and so forth, human-specific diseases in particular. There are agricultural applications, green chemistry, and heritable diseases, which some people would put at the top of their list. Okay, so let's talk about perils, a different alphabetical order. And this is probably not comprehensive as well, but I've tried my best. Since people have sometimes paint scientists as being enthusiasts, heaven forbid, that if we talk about something like on the previous slide, in as balanced a way as we can, then the takeaway is, oh, they must want us to have enhanced babies right away. And so anyway, so this is not just about safety and efficacy, which is, it's also about affecting the unknown. We have to be careful that commercialization will result in something which even though we're voting for it with our wallet, we are not necessarily doing it in the most intentional manner. There's discrimination, which exists despite laws like the Genetic Information Non-Discrimination Act of 2008. There still is discrimination, although there are relatively few examples of pure genetic discrimination based on the DNA alone. There's the issue of equitable access, and that's what I'll start on as soon as I'm done with this list. There's future generations. Are we consenting them properly? Are we on a slippery slope where we start changing future generations and we can't get back up the slope? There's the whole issue of eugenics. Are we really doing the same thing we did back in the 20s through the 70s? In the United States, we forcibly sterilized 70,000 people without their full consent or informed consent. So there's the eugenics that's always hanging over our head. There's hegemony, which is not only could this be used in a military sense. This was raised by James Clapper recently as in a section on weapons of mass destruction, which I don't think quite qualifies for yet. And then there's informed consent. So let's talk about equitable. One thing we can do, and I'll try to say what the problem is and what the possible solutions are, the problems with the solutions, and so on. there are a few examples where we're getting closer to equitable distribution. The original cell phones, which were the size of your head or bigger, there are now billions of them, 7 billion of them worldwide, not quite equitably distributed. But smallpox is equitably distributed. That is something that everybody gets whether or not they can afford a vaccine because it's extinct. The genome sequencing is probably even more exciting or alarming, depending on whether you are predisposed to promises or peril. It has come down in price, I think, even more than editing has. It's come down about 3 million-fold in this short period of time. Maybe most of it happened in a 6-year period. But so has synthesis, and reading and writing DNA in general has come down. And I will submit, I mean, there are many contributing factors, but I will submit that this is not because we bought rooms full of machines. It is what was happening within the machines. It wasn't the parallelization or serialization, it was the multiplexing, where, as Edison did in the 1800s, he sent 4 messages on one telegraph line. You can do millions or billions of reactions in a single tube or a single flow cell. That's called multiplexing, and I would say that the credit and the blame for a lot of this has to go to multiplexing. Still on the topic of equitable distribution, we get all excited about gene therapy, including gene editing, but we need to face the fact that genetic therapy for a while is going to be the $1 million drug. It is the most expensive pharmaceutical ever, $1 million for a dose, and that's because the denominator is low for many of these things. That is to say, the number of people that you divide, the fixed cost of a big clinical trial, and a small number of people that will benefit. That will change when we start looking at diseases or applications that involve many people. Ideally, if the denominator goes up to 7 billion people, then you've got a real blockbuster therapeutic. So what disease would impact, before I say that, the alternative to this expensive $1 million drug is genetic counseling in some cases, and there are exceptions. But that is underutilized. I mean, I think very few people know their own genetics, whether they have carrier status, which is the thing that would impact the 5% of children that are born every year with a very severe genetic disease. Just audience participation, how many people, since you came here, I mean, you could be on the internet, so how many people here know their complete genome sequence? Okay, that's a typical response for a well-educated, well-heeled audience. For the people out in Facebook land, the answer I think was 0, except for me. And as I showed in the previous slide, it is only $1,000, including genetic counseling. So anyway, how do we get down the cost of therapy the way we've brought down the cost of sequencing? And for those of you who've lived through the therapeutic EROM's law, which is the reverse of Moore's law, instead of going, instead of coming down in price, it goes up exponentially. This is, you have to bring up the denominator. You have to have more people benefit. So the disease, what disease does it affects everybody? Once we cure poverty and infectious diseases, we're left with diseases of aging. People mostly die in industrialized nations of diseases that do not kill 20-year-olds. And so I'm not going to go into great detail here, but I do want to thank all the people on the slide, including Pedro de Magalis, who really started our lab seriously on aging many years ago, and continues to have a aging gene database, which is world-class. And then there's a lot of effort on delivery of gene therapies, including these aging gene therapies that Noah Davison and others have worked on. You'll see that the topics that we've covered are going gray, and so once they're all gray, then we have lively question and answer. But you could interrupt if you want. Discrimination, genetics versus eugenics. And now after the colon on each of these things, I'm listing precedents such as they are. So we're going from issues to precedents. Now the precedents are analogies and imperfect, but they're nice to think about. What we have with genetics, eugenics, and discrimination is that we have precedents in mate selection and employment, where you're making a decision based on something that is imperfect predictor, like how well you perform in an interview. But it's not based on genetics, typically. That's against the law in the United States. So what do we have as the alternative to gene therapy that I alluded to earlier, which is, in the case of Tay-Sachs, we have a revolution that occurred that almost unaffected by high-throughput sequencing and gene therapy and so forth, which is genetic counseling. In certain communities that have a high prevalence of inherited diseases like Tay-Sachs, there can be counseling that can occur natally, preconception, even premaritally, where you are informed as to who in the community would be suitable for dating. So you don't even run the risk of having to use abortion later on in life, or in vitro fertilization, which is not entirely benign to the mother's health or convenient. So anyway, there is a very big difference between the communities that have embraced the Tay-Sachs screening, genetic screening, and the sickle cell. In principle, they're very similar, but they have had different outcomes because of the community counseling. That could be considered eugenics, but it's made at the family level rather than the government forcing sterilization. Let's talk about informed consent, and it has some little relationship to the previous topic. We increasingly, in order to keep this revolution on track, equitably distributed and so forth, we need a way of sharing data and cells. I would say right now we are not technologically limited so much as we are limited by our ability to share broadly. Many of the things that we call sharing are actually sharing within the club. If you're in the club, then sharing is seamless. But if you're not, then you don't see it. And that also affects who is studied and how they are consented and so forth. So the non-ideal way, and how many people here have read this book about Henrietta Lacks by Rebecca Skloot? Now, okay, that's the control for whether you've had your genome sequenced. A lot of hands went up on that, and I'm glad. But I must say that after this book came out, to quite a bit of critical praise and discussion, it did not stop genomicists, fortunately not my laboratory, but other genomicists, from continuing to abuse this poor family. And I've met with members of this family, and they were not happy with many biomedical researchers making profits off of this. And then in 2010, they sequenced the genome of Henrietta Lacks via the HeLa cells, which exposed the entire family to some risk, admittedly small, but the issue is whether you're listening to the critics or whether you're tone deaf. So anyway, this happened both in Europe and it was handled a little bit better in the United States. So that's probably not the ideal way to share data and cells. A slightly better way, and this is of course a self-serving slide about the Personal Genome Project. This is one where we do share, in fact, it may be the world's only fully open access, not something where we claim fully access, but it's really like Wikipedia, where the data can be shared completely freely without, you don't need to contact us, you don't need to collaborate with us, you don't need to acknowledge us. You can use it however, you can make a profit from it without cutting us in on it. It is completely open, just like Wikipedia. And that is achieved by making sure that everybody participating in it takes an an entrance exam, a very simple multi-choice thing. Rather than signing a consent form that you don't understand, that's 10 pages of legalese that protects the institution rather than the individual, you take an exam that shows that you know that it is likely that if you're in medical research, your data can escape, just like WikiLeaks and many other escapes, or be hacked. And if it escapes, then it can be re-identified. If there's enough data in there to do precision medicine, then there's more than enough to identify you. So this is now approved by IRB or the equivalents in multiple countries, in the United States, Canada, United Kingdom, Austria, Korea, and China. And some more are in the works. And so what this allows us to do is not only have these open data sets, but to have cells and stem cells that are shareable and all kinds of integrated the way we would want precision medicine to be integrated in the future. So you can actually tell all about people in deep depth, like here's functional magnetic resonance imaging of my brain. It may look like it's serial sections, but I assure you it's just virtual sections rather than real ones. And then here's some of the stem cells we'll talk about in a moment. Now, I'm just going to quickly go through 3 examples of people, and you'll notice I'm using their real names. In some cases, they're personal genome projects. In some cases, they just felt like many patient advocacy groups feel the problem is not privacy to them. It is getting their physicians and scientists to share the data that comes from their body. So these are the Beery twins who could barely stand up or focus their attention when they were young. They were misdiagnosed with cerebral palsy. After a long medical odyssey, when they finally sequenced their genome, it became evident that they had a problem with their SPR gene. And remarkably, the 2 drugs that they needed were actually just over-the-counter nutritional supplements. It's not clear why these are, or it's not quite clear why any of the nutritional supplements are on the shelves. But anyway, this helped. This helped these 2 kids, and when I last met them, they were healthy, engaged teenagers, maybe a little bit more engaged and healthy than you normally expect. So they were terrific. Second example, John Lauerman is a journalist who is in our personal genome project. He presented as normal, but actually in his medical records there were scotomas on his retina and leg pain that they had attempted to do genetics. He had his physicians by guessing. That was the way we used to do it a long time ago. Is we used to guess at a gene. In this case, they guessed factor V Leiden. They guessed wrong. It didn't mean that he had no genetics. He had actually a JAK2 mutation, which is a somatic mutation. So he takes aspirin for life now. And the third one, hopefully you've heard of, but it didn't have the tipping point effect that I expected, that everybody would rush out and get their genome sequenced like Angelina Jolie. But she did, I think, fairly well communicate the idea that she did not have breast cancer. She did not have a mammogram positive or a lump. She had a risk factor, and that's a big difference. And she went from, her physician said, 87% to a 5% chance of cancer by having preventative surgeries. That's not something you want to do lightly. So, on the topic of not doing something like that lightly, let's talk about safety and efficacy. This is not the only thing that is on our list today for perils or ethics, but it is the thing that the Food and Drug Administration will address if we allow them. It's all about safety and efficacy. So from the standpoint of safety and efficacy, what's wrong with CRISPR? We haven't mentioned CRISPR yet. I try to avoid mentioning it, but it needs not one, but 3 different custom macromolecules if you want to hit a completely arbitrary site in the genome. Now, there are ways that you can use any of these things. But typically, you need to design the RNA, the DNA, and the protein. It is hard to deliver. I've already mentioned the AAV is one of the delivery mechanisms. It is inefficient once delivered. There are off-target errors and on-target errors. You can say, how can there be on-target errors? But there are. In fact, that's probably one of the worst parts about it. is expensive. I already talked about that. There is toxicity of DNA damage. We have a paper coming out on that soon. It's hard to handle large DNA inserts. Just it's good at making like random messes, small messes, but putting in a large change is hard. And there's a recent drawing attention to something I think many of us knew, which is there's immune reaction to anything foreign. Other than that, it's perfect. This is an overview of gene therapy in general, and the classic gene therapy, which caused some deaths back in 1999 and 2000, was an additive. You add a gene, it may land in the wrong place, it can cause immune reaction, but that's the main, and those are now approved. There are some gene therapies that are either approved or well on their way to being approved. There's 2,400 gene therapies that have been approved for clinical trials over the years. Subtraction is what the editing, the so-called editing, but I would differentiate between subtractive editing, where you kill a gene, and precise editing. But they're both in principle in the realm of editing. And then finally, there's epigenetic, where you don't technically change the DNA itself, you change the doodads on top of it, the methylation and the proteins and so forth. And the real problem with the on-target is this trade-off between non-homologous end-joining and homology-directed repair, which is the perfect editing. So we can divide. It's not just about CRISPR, which is in the lower left here. There are 8 editors at least, and the number will grow almost certainly. And we can divide them into those that make a mess on the left and those that are capable of precise editing on the right. It's about half and half. Or you can divide them into what part of the molecule does the recognition of the genome that screens through the 6 billion base pairs in your genome and finds the needle in that one place it wants to sit and edit. That can be done by using the DNA or RNA, which are Watson-Crick recognition. A little bit harder because you're recognizing, using a single-strand molecule to recognize a double-stranded molecule. Or protein. There are protein-DNA interactions, which are idiosyncratic. So those are the 2 ways to classify this collection of impressive tools, most of which we got by basic research studying microorganisms. Almost every single one of these came, at least part of it came from a microorganism. So you might say, well, why isn't everybody using these things on the right that allow us to do precise editing where we can do whatever we want? We're not limited to addition and subtraction. And so I will give you an example where we are using CRISPR-free precise editing. And here is, we're achieving a very interesting goal, which is or 3 goals. One is to use non-standard amino acids to have biocontainment, and the most interesting of the 3 is multivirus resistance. You can in principle make an organism or a cell resistant to all viruses, even viruses you've never studied before. I think this is a very profound concept, by changing the genetic code. And you don't have to change that much, possibly as little as 1 tRNA, which means all of the codons that correspond to that tRNA genome-wide. And this is work from these 3 papers. Nili Ostrov she's currently leading this project in our lab. And we've completely changed and tested a genome where 321 changes were made. We're almost done with one where 62,000 codon changes are made, and these are in E. coli. And then we're beginning doing pig and human, where there are 390,000 codons that need to be changed. And the only reason you should believe we can go from 62,000 to 390,000 is that we're on exponentials, and of course that's not a guarantee. So why is it that we think we can get rid of all viruses? Well, first of all, with a 321 change, 1 codon, fairly wimpy codon, it did confer resistance to 4 out of 6 viruses, not quite all, but that's because it was in the upper left where we were changing this wimpy stop codon, which hardly affects any of the open reading frames. So it's up there in the 96% of the viral genes are still translated. But if you get down to the lower right-hand corner, just changing 2 leucine codons or 2 serine codons, and now all the genes in all of the viruses are impacted very seriously, with so many mutations be required to escape that it is, I'm willing to stick my neck out, escape-proof without human assistance, which is, we'll get more into that. Okay, so let's shift a little bit and talk about not so much virology, but what we can do, what is being done, what's sort of some of the first therapeutics that are being approved right now where we use gene editing. And I must say that gene editing is not the star here. The star here really is this chimeric antigen receptor, or CAR-T method, and that is, in a way, additive. You're adding in this strange hybrid between T and B cell receptors. But it's a really great supporting role. The gene editing is a supporting role because what it does is allows us to get rid of additional T cell receptors on these T cells that are attacking the B cell cancers. It can also make it so that you can have universal donors. You could, for the first time in history since the dawn of transplantation, you can now engineer either the donor receptor. In this case, you can engineer the donor so it's compatible to the recipient, and that is done by changing the HLA type to HLA-E, which getting rid of the ones that we normally screen for transplantation compatibility. This allows a much broader acceptance. And you can also make it compatible with these programmed cell death proteins, which is a great output from the Harvard community and others. And then finally, you can make the T cells resistant to simultaneous chemotherapies that you might be bringing in. Here are two categories here. So the idea is the chimeric antigen attacks the B cell cancer, and all these other things make the T cells more compatible with the environment. Now, this is a big deal that we can now have universal donors, potentially. I mean, these are getting approved, and we need to be cautious. There's the peril. But imagine that we finally have gotten to the point where we can make a cell compatible enough, not just changing the HLA, but many other things. And here's the list in the upper left here, some of the things we're changing. We're humanizing in various ways, having the immune system. The sugars on the surface were a big deal, complement and clotting. But one of the things that set back transplantation from pigs to humans, which have about the right anatomy and physiology, was the presence of endogenous retroviruses. And this, when we sequenced a few pigs, we found that that there were 62 in the strain that we wanted to do initially. That was a daunting number for CRISPR at the time. We and others had done 1 or 2 genes at a time. Doing 62 at once seemed very hard, but we did it, and it turned out it was embarrassingly easy. I'm not to downplay the team effort that went into this, but that particular step was 14 days in the incubator, plus a little PCR. Then we had to redo it, that whole thing again with not immortalized pig cells, but pig cells that were capable of doing somatic cell nuclear transfer. And we redid that and then published a second paper on not just getting pig cells in culture, but piglets who are now adult age and reproducing normally, as far as I know. And this is the work of Dong Yu and Luhan Yang and their coworkers now at eGenesis. And one of the things exciting to me about transplant from pigs to humans is not simply this solving of this gigantic worldwide transplantation crisis. It's the possibility that we can do something that's slightly enhanced. And in general, we wouldn't want to, or it's hard to get to justify doing enhancement on people directly. But if you're desperate for an organ, you want that organ to be the best it can be, and you can work out the bugs in the pigs, so you can make them somewhat resistant, or maybe completely resistant to certain pathogens, to cancer, to senescence, cryopreservation, and this will make them in a certain sense better. This raises issues, of course. Now this is epigenetic reprogramming. I listed that on the 4 ways of doing programming. And this is endothelial. This is turning from human stem cells on the left to endothelial, and on the right into neurons. And this can be done in such a way that they have exactly the same media. So we're not using the media to cause this epigenetic change. We're doing it at the level of transcription factors. And Alex and Parastu here deserve a huge amount of credit for making this work. And in collaboration with Jennifer Lewis's lab, Mark Scott, we've put those two together, the endothelial and the neuronal cells, and a variety of other brain-related cells, so that you can now get, for the first time, highly vascularized organoids. These are small versions. They were limited to less than 1 millimeter because the core would go necrotic because they weren't properly vascularized. Now they're vascularized. And that's what these little red tubules, they actually don't, they're not just interspersed, they make tubules like capillaries, and you can see those red ones, the V cadherin on the left. We can also make oligodendrocytes that wrap axons in A with myelin in M, just the way that they do in vivo. This raises another set of issues having to do with, are we making synthetic embryo-like entities or brain-like entities without the rest of the embryo? And we raise this, like many of the issues that I'm raising here today, in advance before the experiments get too far along. Future generations, slippery slope, doing nothing has its own risks. We often want to regulate by taking steps where there's a huge step, a quantum leap, like gene germline versus not germline. But that isn't where we end up. We sometimes have to regulate on a continuum. And I give as an example in this New England Journal of Medicine article I just wrote that we have continuum like for cars, speed limits, blood alcohol limits, and age limits. Here's something where we can impact human health without impacting humans directly with these gene drives. Again, we brought out some of the perils in advance, some of the solutions to those perils by localized gene drives. This is work of Kevin Esfelt and Andreas Speidler. They're shared student with Flaminia Catarucci at the School of Public Health. John Min and Kevin Esfelt and others have gone ahead with these local gene drives. Chuck Noble has done some of the theoretical work as a graduate student on this. And there are many applications for this. And unfortunately, I'm going to try to wrap it up so we get to your questions. There are ways of reversing some of the desert formation, the desertification that has occurred both in Arctic and warmer deserts. Sometimes forests are part of the problems to global warming. And ironically, it may not be entirely, it may not be the fault of us today. It may be the fault of things that we did 15,000 years ago that needs to be addressed. We can't ignore things that are ongoing that have nothing to do with current human effort. And then the final topic that you've all been waiting for is germline, and this affects the unborn. It could be impacted by commercialism. There's already books on the baby business and the eugenics again. We have to be cautious about dismissals and bans. Bans sometimes cause acceleration. I've lived through, up close and personal, many of these bans, recombinant DNA, the Genome Project, stem cells, test tube babies. In almost every case, they increased the curiosity, increased investor curiosity, and often caused billions of dollars to be injected into something that we were trying to slow down. And there are Sometimes the reasons that are given for rejection are weak on examination. We need to be careful about this, is they'll say there's no market interest. Well, there can be as soon as it becomes safe and effective. The yuck factor disappears as soon as 1978 Elizabeth Brown was born. It was no longer the test tube baby in scare quotes. It was actually something that was unethical to prevent people from having their own children. It is very incautious to say that technology is very far away. I've given you some examples of things arriving way, way ahead of schedule. So the sequencing was supposed to be 6 decades away. It was 6 years away. And the waste of money depends on it actually costing money. The NAS report, about 2 years in the making, came out this February. And it, to some people's surprise, came out fairly positively in favor of heritable germline genome editing. Can be permitted under certain circumstances, and they were very cautious about engaging lots of different people and putting limits on this. But overall, it said there were easily conceived circumstances under which this would be possible. And I just want to give my favorite example, which was not that prominent in the report, which is the case of gamete precursors rather than engineering embryos. It's normally framed as you're engineering embryos. This doesn't work technically, and it causes mosaic variation in embryos. The alternative is if you do, say, sperm or egg stem cells, you're not putting in embryos at risk. The standard of care right now is you have prenatal testing and termination that affects 25% of children in families where they're known carriers. And if you do in vitro fertilization, that has impact on the health of the mother, and as well as even more embryos put at risk. So a huge fraction in the United States and other countries are not comfortable with putting embryos at risk, and whether we agree with them or not, it is important to respect them. And so these alternatives should be considered, not considered. And right now there is a block on the FDA for even considering them. There are protective alleles. I'm not going to go through this list. We can talk. It has been discussed. This is from my website. The link is right there. But there are things that you find in natural human populations that make people naturally resistant to certain threats, environmental problems. These are not without, there are trade-offs for these things. An example, fairly famous example, is HIV. You could, by knocking out the CCR5 receptor for the HIV, then you can make a therapy. This happens naturally. The people are resistant to HIV, and it can be done by editing. You can make them, even if they have full-blown AIDS, you can make them resistant to using their own cells to make them resistant to HIV. And that list that I showed, this is the second half. That list goes on, and it bleeds into a set which are not that common in natural populations, or are completely synthetic. We changed from natural population genetics to synthetic biology, and most of these have been tested in animal models. Some of them are already on the way to gene therapies for neurodegenerative diseases, which are particularly significant in our aging population. And the caution here, the peril, is that some subset of these might not only be useful for slowing down or reversing age-related cognitive decline, but they could be used in a positive sense, an enhancement sense, for enhancing in people who do not have cognitive decline. So that's it. Hopefully I've provoked, and if anybody here wants a super big dose of what we just got, felt you got cheated, you had to stand up or whatever, I'm teaching an entire course on this called Biophysics 242 starting January 23rd. So thank you very much.